Guys, so this section in your book is on the endomembrane system. In biology in ninth and 10th grade, you probably learned about a lot of these organelles by themselves. But in this class, again, we're going a little bit deeper with the information. All of these organelles are going to work together. So let's take apart endomembrane. So endo meaning within, and then membrane just has that phospholipid bilayer that we've talked about before. These are going to be organelles that work together, and they're going to be moving things into and out of the cell. So the first one we're going to talk about is the endoplasmic reticulum. That's called the ER. We have the rough ER and the smooth ER. We have the Golgi body. We have lysosomes. We also have other types of vesicles that we're going to see with their own membrane. And these interconnect and work with each other through having their own membrane. So the big important thing here is all four of these are going to have their own membrane, and they're going to work together. Our first organelle within the endomembrane system is the endoplasmic reticulum. This is just a structure of membranes found around the nucleus, and they're actually connected to the nuclear envelope. These have two parts. You have your rough ER and your smooth ER. Technically, your rough ER is more connected to the nucleus than the smooth ER. You can see here the location of the rough ER. The reason why we call it the rough ER or the RER here, you can see, is that ribosomes are attached to this part of the endoplasmic reticulum. When scientists were first observing these organelles, they found little bumps, and they were looking at them under microscopes, and they said, "Hey, these look like it's something rough here," and they figured out that they were ribosomes later on. So this rough ER and the smooth ER, they are interconnected, and again, they have their own membrane. What happens here is you have, in the rough ER more specifically, you have protein synthesis, while in the smooth ER, you have more lipid synthesis. Another aspect, again, we talked a little bit about the function of the rough ER in the last slide, but another function within that rough ER is going to be folding the protein. So there's going to be chaperone proteins here that help fold proteins into their correct structure. We talked about how important protein folding is in the last chapter when we talked about the primary, secondary, tertiary, and quaternary structure of a protein. A lot of that folding goes on within these ribosomes and within the vesicles as well. So you can see here again, it just says the membrane covered in ribosomes for the rough ER. Smooth ER is again going to lack ribosomes. They're going to produce more lipids, and they also help break down some glycogen. And then they start sending what they have within their the rough and smooth ER out towards the Golgi body and other parts of the cell. So this is kind of also like our shipping department, where it's going to start sending some stuff throughout the cell. Specifically, they're going to send the vesicles to the Golgi body. Now, I use the term vesicle a lot in the last couple slides. A vesicle is just a membrane-bound kind of sphere within the cell. These organelles, again, they contain materials and things the cell is going to need, and they kind of like they're kind of transporters. So, like, think of like a UPS truck here. So the UPS truck is going to take things from one facility to the next to the final destination, which might be your house. So you can see here we have our nucleus, our rough ER. Here's where we're at within the endomembrane system. We have these vesicles that are going to start transporting things into and around the cell, and sometimes even out of the cell. Specifically, these guys go from the rough ER and smooth ER to the Golgi body. The Golgi body we're going to get to here in a second, but the Golgi body is like the UPS center. It's going to be where we really send out and ship out things. Now we arrive at the Golgi body or the Golgi apparatus. You're going to hear both of those terms used pretty frequently, and they can be interchanged there. So Golgi body and Golgi apparatus mean the same thing. What these are, they look like flattened vesicles here, and they're just like a department where you're going to start like intaking the vesicles and folding them even more, and they're going to start sending them out. You do have a cis and a trans face. You really don't need to know what those means. You just need to know that that's where there's one side that's going to be where the incoming vesicles come from, the smooth and rough ER. They're going to be folded. The vesicles are actually going to be they're going to be fused with the membranes here. Then the proteins are going to start being folded. Other lipids and macromolecules are going to be tagged, and they're actually going to be shipped out on the trans face or on the opposite face of the Golgi apparatus. So what this is just like a big UPS center. It just takes things in, it organizes them, says, "Hey, I need to send this to this part of the cell, or I need to send this out of the cell," and so this is just like a shipping center of the cell. This is a great image of the endomembrane system. Again, a lot of times in college prep bio and 9th and 10th grade bio, you guys learn these organelles independently. What you need to know from this section is that these organelles work together. It's not that they're free-floating and free-working organelles. These organelles work together. So you can see the rough ER here is going to start sending vesicles to the Golgi body. The Golgi body is again the shipping center of the cell. It's going to package, tag, and move things around and send those vesicles out to different parts of the cell, either inside the cell or outside of the cell. Again, love this GIF or this animation here. You can see where the rough ER here and the smooth ER is going to start sending proteins to the Golgi body. The Golgi body is either going to send them out of the cell or to other locations like the lysosome, which we're going to get to here shortly. All right, one of my favorite organelles is the lysosome. Now, this is technically included within the endomembrane system, but it's not the end goal. It's not the end location for every single protein that's going to be made within the rough ER or lipid within the smooth ER. What happens with the lysosome, these guys are like our custodial staff of the cell. They break down worn out organelles, worn out proteins, things that the cell doesn't need. And they do this by having digestive enzymes within the membrane. Now, lysosomes are pretty interesting. We'll talk more about the enzymatic functions of these digestive enzymes later on in the next couple chapters. But these guys are like a recycling center of the cell. We will reuse some of the proteins and amino acids that break down in the lysosome and other parts that the lysosome breaks down. So this is a pretty efficient way of kind of recycling things and not really wasting things in the cell. Another important organelle that's very closely related to the lysosome are peroxisomes. Lysosome is a more general term for an organelle that just breaks things down. Peroxisomes are more specific. So what peroxisomes do is they help break down alcohols, formaldehydes, and other dangerous elements and chemicals that are going to be found in the cell. One of the ones that I really try to focus on is the free radical oxygens and hydrogens that are found in the cell. Through respiration, cellular respiration that is, and other processes, you're going to have these free radical hydrogen and oxygen ions. Now, they don't seem like they're that bad of a thing to have around, but if the cell accumulates so much of one of either, they can become pretty deadly. So what the peroxisomes do is within their membrane, they have these enzymes that actually convert the free radical hydrogen ions and these oxygen ions into hydrogen peroxide, H2O2. That looks very close to water, and yeah, it is very close to water, but remember, structure determines function. This doesn't mean that it is water. It actually has very opposite properties of water. Where you need water for life, you actually die when you have too much hydrogen peroxide within your system. So what the peroxisome does, and you're probably thinking like, well, why would it take hydrogen and hydroxyl free radicals and make them into hydrogen peroxide? Well, they have a secondary enzyme in there, and what that enzyme does is catalase. It takes the H2O2, the hydrogen peroxides, and breaks them down further into water and oxygen. So it's kind of a two-step process where you're taking the hydrogen and the oxygen free radicals, making them, yes, something more deadly, but then you take that deadly thing and then you break it down into water and oxygen. So this is kind of a two-step process to remove these hydrogen and oxygen free radicals.